Okay, welcome everyone and welcome to the first SSA ACT meeting of 2026. And as usual, I would like to start this meeting by acknowledging the traditional custodians of this land on behalf of the Statistical Society of Australia. I would like to acknowledge the traditional people living here, Gangwal and Gambia peoples and their connections to land, city and community, and we pay our respect to the elders past and present, and extend that respect to all the Aboriginal and Torres Strait Islander peoples, people who might be present here today. And welcome everyone again. My name is Luca Maestri. I'm the current president of the SSA ACT branch here in Canberra. Welcome to those online as well. We are here at the ANU in the College of Business and Economics, and we have already listened to the talk that Angus Macford has prepared for us. So Angus is an academic from the ANU, specifically from the National Centre for Epidemiology and Population Health, also known as NSEF here at the ANU. Angus Macford is a mathematical modeler of infection diseases, and his work on projects across a wide range of diseases and pathogens with a specific focus on foodborne diseases. And he's developed and maintains an art package called Pool Test in R for applications where data are tested in pools. And in fact, the main topic of this talk today is about statistical strategies based on tests for pool data. Yeah, thank you, Angus, for being here today.
Okay.
And we'll keep questions for towards the end of the talk, let's say.
Yeah.
Okay, thank you.
Is that the format of entertaining question? The end walk through that.
It's okay. That's fine.
Yeah, it's fine. So, yeah, thank you for the introduction. Thank you for the introduction, Luca. Yeah, so I'm going to be talking about group testing and why one might do that, what kind of applications it has, a little bit of, and then talk about some of the work that I'm doing again, looking to improve the design of the surveys using group testing. So, first of all, let's see if we can change those. There we go. What is group testing? Well, it's also known as pool testing. I think it's a slightly more informal name. I think the main problem with pool testing is the name. If you Google pool testing, you get information about chlorine. Yeah, and it's not very. I think back pool testing is sort of that. But intuitively, pool testing is a better name because the basic idea of group testing is unlike individual testing, where you're testing people or biological samples individually for the presence or absence of the disease. In group testing or pool testing, you pool together the samples first, and then you can test the pool or group sample. That's all it is. That's example. So that's what we're going to talk about today. So a bit of a sort of a history. So that's the earliest example of group testing. Useful for a wide scale is something called the Dorfman procedure. So it was, and it's one used to get. So Robert Dorfman was working with the US military in World War II, and they had the big problem. They wanted to screen all new recruits or potential new recruits for syphilis, but they had a lot of people they wanted to screen, and the test, the blood test that was involved was very labor intensive. So they were testing for presence of a particular antigen, which would indicate that they're infected with syphilis. Now, so unfortunately, they had someone who knew some stats working with them, and Dorfman suggested that instead of testing them individually, you group them in pools, and this as a way of screening everyone to massively reduce the number of tests. So the basic idea is you take groups of, in this illustrative example, four people and pool their blood into one test tube, and another set for another test tube, another set for another test tube. And then you test those pooled samples. And if they're negative, well, we know that all the people, and by negative you mean there's no antigen present in the blood, the pooled blood sample. That means that none of the people who contribute blood to that pool are infected. But any time someone that pool that does have evidence of antigen in it, then you go back and retest all those individual 4 people who contributed blood to that pool and check which one of those people are infected. So it's fairly simple, clever idea. So we go through and need to do 12 individual samples for these 12 people. And we now need to do three tests on the full bloods and another 4 tests on these individual bloods from this group that tested positive. Now, so again, it took us 4 and 7, which is 12. So right where we use fewer tests to screen exactly the same group of people. So the obvious problem here is that this is not always going to be fewer tests, right? Like what if this particular set of 12 soldiers are unlucky and 3 of them are syphilis, and not only that, those 3 end up in different pools. In that case, if you're screening 12 people, you do 3 group tests, and then you go back and individually retest everyone. So you're still doing 12 individual tests plus an additional 3 group tests. So you're now doing 15 tests to screen 12 people. So that's not bad. So obviously you want to optimize this, and I mean, Jordan, as I said, was using statistics. So that's immediately what we did. So the group size is what we have control of. The number of blood samples that we put into our tubes. And we can choose this group size S. The prevalence, that is the proportion of people in your population who have the antigen in this case, we can't control, but we might know it. We might know in previous tests, roughly the proportion of people who test positive to test immunity. What is not known and sort of randomly is the number of tests, the number of tests to do. So the idea is we want to minimize the expected number of tests that you have to do to screen on average per sample, right? So individual testing is 1 test per sample. We're trying to beat that. Before I go on, I was informed that this should not have many equations. These are wonderful equations. So it's sort of this is and the equations are first optional for this talk in terms of understanding what's going on. But basically, the idea is that if in the case where all the blood samples which go into a pool are all negative, then we don't have to do individual retesting. And that's why you say that one test if they turn out to be negative. If they turn out to be positive, you haven't saved them, test they have to do more. And so you're balancing these 2 things. If you want the pool size to be big enough, so that you say, well, tests and they're negative, you don't want to be so big so that they pretty much guarantee to be positive every time and you always be tested. So, and here's the balancing act. So the expected number of tests that you have to do is, well, 1 for the pool plus S tests every time the pool is positive. And what's the probability that the pool is positive? Well, it's the 1 minus the probability that they're negative. So, and this one here. So for small theta, this is approximately 1 S plus S times theta. And it turned out you can, just fairly simple algebra. This is minimized when S is approximately square root of p. So for instance, if the prevalence of syphilis is 1% in its population, then a pool size of about 10, then that'll be helpful. And you end up only needing 20% as many tests as you would have to do if you did individual tests. So you're reducing the number of tests about 2 by 8%. So it's a math reduction. So and in general, the lower the prevalence, the bigger the pools you can use, and the bigger the pools you can use, the more costly things you have. So this is obviously not just used during World War II; it's used all the time today in various infectious disease programs. So perhaps the biggest growth in most recent years was during some of the early years of the pandemic, when there were enough people being infected, enough people that we were screening lots of people, testing lots of people for COVID, but there just simply weren't enough tests in Australia or in many countries to test everyone individually, even if we wanted to. So there was a big pressure to reduce the number of tests to screen the number of people. So this kind of Dorfman procedure was used often. But it's often used in screening for rare diseases, screening for rare pathogens, rare tests, and kind of something like that. So that's the Dorfman procedure. Now, if we see the I'm sure we can think of lots of variants of the Dorfman procedure, which might in some cases improve efficiency. And since 1943, people have done a lot of that. It's talked about lots of different variants, which can improve. So I'll just talk quite briefly about a couple of them. Because I think they're interesting. I think they're sort of quite fun ways to actually make it more efficient. So one is to do this in multiple stages. So you start with the same, say 4 people in one screen. Instead of immediately putting all their blood in one pool, you put them into 2 small pools and then combine those. And then you start your testing with the biggest pool and work your way backwards. So if the biggest pool containing a little bit of everyone's blood is free of the pathogen, well, all 4 examples are good. If the big master pool is positive, then someone somewhere back here is infected and you're trying to figure out who. So you work your way backwards, you test the intermediate size pools, and if they're all clear, then those people are clear, and if they're not clear, you keep on going to do individual testing. So again, a simple change, looks like a simple change to the process. But again, we can cut the number of tests to do it half again. So compared to you can just bring it up normal. So for 1% prevalence, you might only need on average 0.1 test per person to screen everything. So it's perfect. So these are folded after all, and they're marked on rows and columns. So we might want to take advantage of that. So one clever solution is to, instead of doing, which can in many cases avoid the individual testing altogether, and avoid this sort of complex if this and that sort of logic, is to pull across to a range of blood samples for people in rows and columns, and to pull across rows and pull across columns. And do individual tests there. So now, if you've got any one infected person here, as you have in this case, you look exactly one column, exactly one row will end up having a positive result. And so from that, you can figure out, I'll ask you this for a person who's got syphilis, we better listen about that. Again, the obvious problem here is if multiple columns and multiple rows simultaneously test positive, then you don't know who exactly positive. It could be that all 4 of these people are infected. It could be that these 3 are infected but others not, or these 2 are infected and these 2 aren't, or etc. So lots of combinations. You have to go back and individually test them to figure out which 2, or at least there's at least 2, but which 2 are infected. So there's lots of variants here. I think my favorite area, and what I'm going to be spending most of the time talking about, is the simplest one. You just don't test. So if all the people, if you contributed blood to the sample are all negative, you get a negative result, that's great. If it's a positive test, you don't know which of these people are infected. You know at least one of them are. And so obviously this is no good for screening. This is no good for figuring out who's infected, so you can, not admit them into the military or treat them or whatever the outcome is that you're trying to achieve by the screening process. But that's not always the goal. It's not always, you don't always need to know exactly who is infected to do something useful with that information. So, for example, if we're talking about we're talking about sheep, we're talking about animals that are being screened in Australia for infectious disease. Sometimes you need to demonstrate that if any of them are infected, you're probably going to apply treatments to the whole herd to protect them as well. So it doesn't matter so much which particular sheep is infected, you don't even know, it's treat them all. So you don't want them tested. Or there might be strict export conditions where if any of it is detected in your sheep herd, you can't export from that herd, or you can't export from the region. So it's really more about detection rather than screening and figuring out which sheep are infected. Similarly, this would be imported flowers and imported products where we're trying to keep pests out any infected or affected biological samples that they're contaminated, reject the whole lot anyway, so we're not keeping that at the border. So the case that I'm most particularly interested in is different from these again. So it's when we're looking, instead of looking at people, where we care about who's sick and who's not, and treating them and helping them, we're looking at the blindness that made these people sick in the first place. So for the vector-borne diseases, things like mosquitoes, the black flies, which spread diseases from people to people. So now I'll talk a bit about that. So one of the diseases that I work on quite a bit at the moment is called lymphatic filariasis. This is a very unpleasant disease for people who get severe symptoms, but many people have no symptoms at all. So the way it works is it's caused by a very small wound which lives inside of, well, hopefully not you, but in some people, inside of adult worms living in the lymphatic system, which is sort of the drainage system for your body. if the lymphatic system is damaged, then fluid can't drain from your limbs and your arms, and from the whole of your body properly, and so fluid builds up. So, and this is why it's called lymphatic fibrosis. So these worms, if you have chronic burdens of these worms, and they live in for a long time, they can. Their lifespan is 5 to 10 years. It's not like 3, but they live a long time. If you've got lots of these worms for a long time, it can cause, in some people, irreversible permanent damage to the lymphatic system, which then means that they're swelling in their legs or their groin. There are about 40 million people in the world today who are disabled because of lymphatic fibrosis, as affects their ability to walk or to work, and there's lots of circumstances around it. So it's caused by these worms, but the worms can't in fact reproduce, do their full life cycle, that is, they complete their full life cycle inside of a single human host, which is their host. They need to spend a few days of their life inside of mosquito, otherwise they won't mature past infancy. They'll stay an infant forever. They don't spend at least a few days in front of a mosquito. They're very, very, very particular. So the way that works is the adults get in the lymphatic system, and they breed excessively, and there's millions of these tiny, tiny worms in your blood, and mosquitoes come and take a sample of your blood and then fly off. And mostly they don't buy anyone else, they go in their eggs. Sometimes they go by another person and then they get infected with these worms as well, and that completes the cycle. So it's horrible disease. But most people who are infected and who have the worms don't know they're infected. It's found in many parts of the tropics and subtropics. It used to be in Australia until the mid-last century. And there's a big global campaign to eliminate it. Now, it's too hard to test everyone to see if they're infected. And so rather than even easily doing Dorfman screening, they just test the current strategy is to treat everyone. Assume everyone might be infected and treat everyone. So that's called mass administration. So there's big global campaigns around the world in fixed areas to do mass administration. Okay, but this means that the standard process doesn't involve any testing. Most people don't have any symptoms. And so, and it might be years and years between getting infected and clinical symptoms. So you need to do some kind of studies. You need to figure out, is this disease spreading? Have we got rid of it yet? Can we stop treating people? And so one of the approaches is to collect the mosquitoes, trap mosquitoes and test them. And that's where the testing comes in. We don't care which mosquitoes are infected. We just want to know roughly what proportion of the mosquitoes infected and at where. So this kind of approach is being used a long time for other infectious diseases. So very quickly, another example is onchocerciasis. Very similar sort of thing, caused by a worm, lives in you for a long time, very nasty effect in some people. This time it's rather than swelling blindness, hence the common name root blindness. And again, but it's still by the same drugs, lives in the same part of the world as lymphatic fibrosis. Often there's more overlap. And so the approach again is to do mass administration, and the way they do surveillance, a big part of that is by catching the flies, the biting flies that spread the disease, and testing them. Okay. So, but what are these surveys look like in person? So they're called it's a big name, molecular xenomonitoring. Xenomonitoring means you're doing monitoring not in humans that you're actually interested in, but in a foreign species, in this case, mosquitoes or flies. And molecular means that you're doing a test. So the molecular xenomonitoring surveys, in practice, they look a lot like a sort of cluster survey that you might do based on household. So, in a regular cluster survey with humans, you might choose an area where you're interested, randomly sample households, one of those households, normal people who, willing to give you blood or answer a questionnaire, whatever it is, and test those people individually. The differences for these molecular xenomonitoring surveys is there's two differences. So, first, well, there's one key difference. Instead of enrolling people in households but trapping mosquitoes, there's no consent involved, so that makes it much easier. And second, where we don't care which mosquitoes are infected, just which house they pick up. And so we match them up with pools and test them. Okay. So, what might be the purpose of these group tested surveys? Why would we do it? So I think it's sort of two broad purposes. One might be detecting presence. Is the disease there, or is it? And one is estimating prevalence. And then in your design, you have to kind of think a bit differently back into it. So, I mean, detecting presence, you might be using this in very low levels. Where it's pretty short of gone. If it's there, there's not going to be very much of it. So we should really know about presence absence. Can we find any affected for black holes? And this might be so you can find locations where you're going to mop up the little the area and treat everyone around it with some bit more mass restriction, a little mini MDA around where you find infected mosquitoes. Or you might be doing it to flip it on head to prove that it's gone. You're proving to WHO we've got rid of the license, we don't have to do mass restriction anymore. Game over. So that's one of the options. But that's not always the case. Often you don't just care about presence absence, you want to know something more, you want to know about the prevalence of the disease. So again, you might want to stratify areas into risk based on the prevalence, either low. You might want to compare prevalence of the disease in the population to some sort of threshold to make a decision. If it's above this, we'll do this very expensive, complicated intervention. If it's below this prevalence threshold, if there's not many infected mosquitoes, we'll just watch and wait. And that's the practical conclusion. Or you might want to compare, use the prevalence of mosquitoes as some sort of measure to kind of measure are our interventions working? So we might want to compare prevalence in mosquitoes before and after an intervention, or in a control and a intervention arm to see if what we're doing is successfully reducing the prevalence of the disease in mosquitoes. So that's why you might do these group test surveys. You might just simultaneously come to go for, if there's nothing there, great, in terms of absence. If there is stuff there, you would like to know a bit more than it's there. You want to decide. So the problem is that in this whole group testing framework, if you're not going back and individually review testing, you don't actually know what fraction of the sample were positive. You don't know. So I mean, in individual testing for Dorfman, where you are individual testing, or other areas where there are group testing, it's quite easy to estimate prevalence. It's just the number of samples, the number of positives, the likelihood is your standard binary likelihood. Maximum likelihood estimate of prevalence is number infected divided by number of total number. You can do like Bayesian stuff. There's lots of conjugate primers, beta, beta is conjugate to monomer, so that works really nicely. So it's all good tested. This is for either individual testing or if you're doing group testing, but with testing of individuals. But if you're doing group testing without retesting, it gets very nice and very quickly. First, in real life, actually, there's not a single tool size. It's not like we're all putting them every. All the folders line up in rows. Mosquitoes don't line up in rows, so you get lots of mosquitoes over here and not many over there. And so you actually get different pool sizes. And so your likelihood still looks like sort of pseudo, binomial, but the probability of a pool being positive depends not just on the prevalence, but the size of the pool. The more mosquitoes in the pool, the more likely it's going to be positive. So it's looks a bit like a binomial likelihood, but a bit more complicated. Maximum likelihood, again, it's pretty straightforward, numerically calculate it, not hard, but there's certainly not a nice clean form which is intuitive for people who to use like statistical programs. Also, the maximum likelihood estimate is often quite high, so often overestimates what the prevalence is, so you need to go approaches. As always, you can always do Bayesian inference, but again, there's no nice continuity anymore. So it's point of the slide to say it gets more complicated. So, and I think more interesting. So to help with some of these problems, I've sort of developed some software. It's not the only software in this domain, but I can talk about software as well if people are interested. So I've developed a package called Pool Tester. This is for estimating prevalence, and I suppose taking that step further, we can not just estimate prevalence, but do sort of on regression modeling. Logistic type of regression modeling is sort of a binary outcome, but you're adjusting the fact that you're not looking individually, but looking at pools and pools of different sizes. Then using that sort of regression framework, obviously you can compare the prevalence of the different samples, you can do hierarchical sampling designs, like we have with our real, remember NX surveys are their cluster surveys, so we need a clustering, which is often quite severe. And recently I've sort of been working to extend it so we can do spatial regression modeling. We can map, we can use it to do mapping of expect a higher resolution speed. I'm also working on another package called Power, which is kind of the flip side of this, not the analysis of the data, but the design. Doing sample size calculations, design effects for designs, optimizing design. So what I mean, why do we need to things to here? If again, like I said before, if you don't retest individuals, you lose all this information. If a pool of 20 is positive, it could be that one of them is infected, or it could be that all 20 of them are infected. You don't actually know. So you actually lose quite a bit of information by testing. And how much information you lose depends on the design. So it is worth watching some design. I'm also developing sort of pointing. So these are both art factors, people can go mind writing a bit of code. Not everyone likes that though, so I've also got an online interface, the point and click style thing, using Shiny apps, which lets people do the basic functionality from both these factors. And particularly these two are sort of, well, it's all work in progress, so they've always added to improving things, but this is, I suppose, the most so. Okay, so I've talked a bit about what the software is, but what are considerations for design? What do we try to optimize when we're designing surveys? So we're trying to identify survey design that maximize information about the object of interest, in this case, prevalence of the disease in your function, in your population of this, the minimum survey errors. And just to be clear, minimum survey can be quite extraordinary in some of these cases. You have to cross rivers or drive through locations to set up the traps or sampling people. And setting up the traps, there's collecting the mosquitoes, there's sorting the mosquitoes, there's testing the mosquitoes. So there's lots of different components to the survey that you're trying to balance getting bang for buck in terms of information. So if we ignore for the moment that most practical surveys of this kind are actually not simple random surveys, but sort of cluster survey designs, we can think about the trade-offs that we get for using impressive. So if we're just doing simple random sampling and then testing people and testing mosquitoes or people wherever they are in groups, we've got one design choice really, which is pool size. How many units are you going to put in each pool? And I've called this is not pool size and size, this is S. The pool size is called S for size. So you've got one choice, pool size, and there's this one key sort of those design factor thing that you consider in design, which is the prevalence. And so, I mean, how do we quantify information from our surveys? Well, from our sampling design, well, for a pool of size S, we can calculate the efficient information, which is on this form. So it's somewhat familiar to the binomial something. So if in fact we don't have a single pool size S, but a whole range of pool sizes, some pools are size 2, some size 10. The information that we get from across those things, assuming that all the benefit is out. So that's not that much more complicated, but it is a complication, which I will mostly do. So this is the information for a pool size S. And from that we can calculate the design effect. Now, how much with full testing we lose some information? We understand that. How much information do we lose? How much more, sampling, how much more tests do we have to do? Or how much more sampling do we have to do, that is, because we've lost information from full. And that's our design effect here. So we look at how much information would you get from S individual tests on the mosquitoes, and compare that as a ratio to how much information you get from one group test, hostile test. So that's our ratio here. And this has this nice simple form, which is it's increasing, the design effect increases, its prevalence increases, and its test increases. And I'll show this here. So here's our design effect here on the y-axis, how much more sampling we actually have to do. And on the x-axis is prevalence of the disease. So our reference is individual testing, so group size, pool size is 1. Right? Every unit is tested individually. So that's one there. And as the prevalence increases, you lose more information, so the design increases, but it increases the design effect grows very rapidly when your pool size is good. This makes sense. If you make it pool size too big, the prevalence is 10%. right? And your pool size of 20, most of your pools are going to be positive. Most of those pools are going to be in that range where it's not giving us lots of information. While the prevalence is very low, for a pool size 20, most of those pools are going to be negative anyway. A negative pool is the same information as testing individually. You know they're all good. So it's the same amount of information. So it's not going to give much information loss. So you get this relationship. So this is how much more sampling you can do, right? If you're doing group testing. But the whole reason we're doing this is that we can reduce the amount of testing you do. So if testing is the hard bit, it's the expensive bit, it's the costly bit, you still need to trade off the effort to do the sampling and versus the effort to do the testing. So I might come back to this. Yeah, but yeah, to trade off the different costs. So you've got two major costs: the cost per unit, the cost to collect one mosquito, cost to collect one blood sample, and the cost per pool. How much does it cost to perform your test on your pool mosquitoes? And for a given ratio, that's going to determine what your optimal size is. So make the pool too big, and you do this to be on the test, get your information. Make the pool too small, and well, you're just doing lots of negative results. You might as well be full bit bigger. So there is a lot of pool size. So for a particular ratio here, such as 4, you get this optimal pool size like that. So this black line is the optimal. So for prevalence of 2%, the optimal is about, what's that, 14 or something like that. So the 14 mosquito per pool. While if the prevalence is higher at 10%, the optimal is about 10. Okay. And this grey line is not confident interval. This grey line is the range of pool sizes, which they're not optimal, they're not bad. They're plus and minus. If we're evaluating a design based on what's the cost of the information, what's the dollars per unit fisher information it's worth. These are all the designs which are only up to 10% in the risk. So there's a range of pool sizes which are okay. So if you're at 10% size pool size of 50, bad. But if a pool size of say 12 works is nearly optimal across quite a lot of your preferences. So even if you don't know what your preference is ahead of time, you can say 12 pool size of 12 is fine. Okay. But that particular trade-off depends on the ratio of the costs. The more expensive pooling is, the bigger the pools you want to do, and the cheaper the pooling is, well, there's no point pooling. You might as well just test everything. That's right. If the cheaper the testing is, you might as well test it. So here we have lots of different cost ratios. So for here at the bottom, we've got the cost ratio, cost of the pool, cost of the test is 0 compared to collection. It's free to test. And it's free to test, just test everyone. There's no point pooling. So optimal pool size is 1. So at the other final extreme, we've got this case here where the cost of pooling, this is the cost per pool, that is the cost of one test, is massive compared to the cost of collection. So this is kind of the reasonable case to consider when you've already collected all the things. That's the sunk cost. It's the sunk cost. Now we've already collected all the mosquitoes. We have far more mosquitoes than we can possibly test. We've got a budget to test for. We really don't know how we're going to pool them to get the most information, given we've got a finite budget. And it doesn't make sense. Again, if prevalence is 1% and there's 1,000 mosquitoes in every pool, you learn nothing. Right? You don't, because they're all going to be positive. So you don't know prevalence of 100% and 50% and 10% or 1%. You can't just differentiate between those cases when the pool size is too big. So there is a limit to pool size after which you start losing information. Okay, and in between, so even for quite cheap testing, even if testing is only 10% of the cost compared to collection, it does still actually make sense to do pooling if you go back a certain prevalence. So for a prevalence of 1%, even very cheap tests, you're looking at pool sizes of what is that, 6 or 7. This is all at once. Actually, that's 1, 2, 3. Pool size of that really big. Okay. But this is all we're looking at simple randomness. If you're not doing simple randomness, if you're doing cluster sampling, and there's the disease is clustered in space, which is often the case, it gets more complicated. you have more design choices for data, so a pool size, S number of units per pool, and how many pools you can do in each location. I can do intensive sampling in a few locations or spread it out. Again, like a photo session before, it costs a lot of effort to go out to the new locations, and so the cost per location can be high. So you might want to keep the number of locations down. And yeah, and if you multiply this together, like how many mosquitoes are we catching, how many people are we sampling per location? S times N. So those are design choices. Design factors, prevalence again, and correlation. How much correlation of my own interest is there out of different clusters? If there's no correlation, if the disease is uniformly spread everywhere, and you might as well just go on forward and say that it's representative of the whole country. But if there is clustering, then you need to go to more varied locations. Again, if we're optimizing something, we need to think about costs. So the costs are cost per unit, the cost of collecting one mosquito, the cost per pool, which is the cost of doing a test, and the cost per location, the cost to actually go out there. And here, for example, assuming that they have this ratio of 1 to 4. And so here we've got, and so again, the optimal really strongly depends on how much clustering there is. So for some of these diseases, it's not unreasonable to think that the cluster correlation, the inter-cluster correlation coefficient would be as high as 0.1, like it is very constant in space. And so in that case, the optimal pool size are quite small, and that's not directly related to somehow pool being making desire. It's just if you've got a big pool, right? You need at least 2 pools per location, otherwise you can never estimate how correlated outcomes are at a location. Right? If you only ever get one piece of information related to one pool, then you can't estimate correlation, so you can never get reasonable confidence intervals on your estimate. Right? So you need at least 2 pools. If you've got at least 2 pools, and they're both 50 each, that's now 100 mosquitoes per location. That's a very large sample per location. And the higher the correlation, the less information you get for each individual extra mosquito or person who is from each site. So so what correlation does is it puts a cap on the maximum number of catched per location, S times N, which then sort of indirectly then puts that cap on what your pool size can be. Anyway, so your optimal pool size is quite a lot smaller. So here before is our optimal pool size, assuming either there's no correlation, we know that correlation, or equivalently that this is a simple random, so we ignore that. This is the optimal in that case, optimal pool size. The optimal pool size, when there's even a little bit of correlation, 0.01 is a lot smaller than your optimal pool size as well. So, which is kind of bad news, means that there's optimal pool sizes are smaller, you don't get quite as much efficiency from the whole point. Again, that changes with depending on how to change the cost structure. So for a particular optimal pool size, this is what we call optimal pool size. Optimal number of pools also change with prevalence. So that the more the higher the prevalence, the more pools per site people. But that doesn't. But the optimal number of mosquitoes per site doesn't really change. There's an optimal number to catch if we're given a cluster. What changes is how, for different prevalence, what changes in how you should group those into different pools. Now, again, this is just for a particular cost structure. If the cost of going out to a location is not very high, say compared to the test, but what if you make everything more expensive? Right? What if testing is more expensive or going out to a site is more expensive? So in this case, we've made going out to the site, going to a location, setting up much more expensive. And then the optimal number to catch per site goes up, the optimal pool size goes up slightly, and the optimal number of pools goes up as well. So instead, we make testing more expensive, right? We make testing more expensive, then the optimal catch size goes up, a bit, but the optimal pool size goes up. And if you could do, let's get that, even though they have components. Okay, so we can further generalize this in multiple ways, and I won't go through them. But here's some sort of thoughts. I mean, for a real survey, right? We don't specify how many mosquitoes we're going to catch. We set out a plan. We get a mosquito, as many mosquitoes as we get. Get what we get, and we don't get upset. We sort of test what we have, and we forward according to what we have. So in real life, the sample size. So how do you design a survey around that? Knowing that the sample size is going to be potentially quite big at this place, and potentially not there. What if there's a variation in sample size? And then how does that interact with falling? Well, it's in complicated ways. So, so again, here the design choice is not so much how many mosquitoes you catch, but the duration of them, like how long are you going to leave the traps out there? And yeah, so I'm still working on this area a little bit, but so in general, it appears the more variation in patch size you have means less information. This kind of in two different extent. Like if you're getting a lot of mosquitoes from one place, if on average you're getting, say 30 mosquitoes from your location, but you're getting a lot from some and almost nothing from others, you're getting the marginal information you get from lots of mosquitoes here doesn't outweigh the loss of information from the few mosquitoes there. So generally, yes, you get even less information from this. So you need to inflate your sample size calculations a bit more, if you're taking into account the fact that you got cache sizes. Some fun thoughts here. So I mean, the good survey design is really quite sensitive to the setting, I suppose, is on my take homes from this slide here. So the degree of correlation of clustering it seems to be quite variable. So in some countries, for instance, in the Dominican Republic, when they do these surveys, it doesn't appear lots of clustering in the disease. It seems to be fairly well spread out across the country for whatever reason. It's quite surprising. It's the only place I've seen this. Everywhere else I've seen people report about the clustering disease. It does really like custom house within town and within neighborhood, even within houses. It's a very sort of focal disease. And so the degree of correlation between these is quite high, and you need to take into account when you're looking at designs. Also, the costs are quite different. So this is a disease which is spreads in urban India, also spreads in extremely rural parts of PNG and in beautiful tropical island countries, and experimented in Australia, used to be in China until the late 8th century. So in all these different settings, the cost structures can be very different. And so your design, optimal design is going to depend on that. So published MX surveys, so surveys which use they pretty much use a one-size-fits-all strategy. At one point, someone decided that 20 size pulls of 25 is good, and everyone has done that ever since, even if it's losing them lots of information potentially. And they don't justify sample size. Again, I suppose they take the approach of you get what you get. You don't get upset and you do a post-hoc, sample size calculation saying, sorry, we'll keep that. But I mean, I think we can do better than that. So, especially in this space, there's guidelines being formed by WHO to inform, how much surveillance needs to be done for these diseases. And similarly, for other, if we're not thinking, taking a step back, thinking about disease control in Australia or monitoring pests coming into the country, monitoring diseases and export animals. This is a space with lots of vested interests and lots of litigious people who have a big interest in proving that their diseases, their countries are free of disease or their flocks are free of disease, and keeping it that way. And they also have a great interest to do with little surveillance that they possibly can. They don't, So there's lots of guidelines around this, but again, they don't. The one that I was familiar with, the one about infectious haven't considered these things about the design and taken into account in what they're looking. One of the key things is, correlation is often unknown. People haven't bothered measuring it, haven't bothered reporting it always, but it makes a massive difference to your estimates. And to your advice. Just very briefly, on things I'm working on at the moment. So the package I talked about, core power, which is the sample design that helps you optimize for the cooling strategies. That's taken into account. I think for those who haven't developed an art package, it's not work, don't do it. It's, you know, if you're considering it, yeah. I mean, we're kidding, that's going to be wrong. Yeah, but it can be a very rewarding site, and it's particularly rewarding when people use it, and people use it for things that you weren't even aware of. So I think the full best art package, almost all of the citations for certain with all these diseases spread by ticks in ticks that all these diseases I didn't know anything about, but fortunately people are also using it for lymphatic filariasis and stuff like that as well. So anyway, developing that package is in process. Working on an automated payment system of outcomes and this type of work, and also working on making these more accessible. Again, full power as an art package accessible to people who have to write their code or to have to use ChatGPT and how it works. But even that is a step too far for many people. So it's very helpful to have this kind of politic so as well. One other thing, one thing that we're going to sort of end this slightly is using regression for the modeling, the sort of extent to sort of mixed picks, Gaussian process regression modeling with this full data to do mapping, to do risk prediction across the country. So here are two maps. I haven't labeled them on purpose. One of them is the risk in mosquitoes, and one of them is the risk in humans across the country. Samoa is fortunately sort of tilted sideways, so you can fix two maps of Samoa next to each other. So this is one Samoa, and this is another Samoa right next to it. One of these is humans, and one of them is mosquitoes. Can you difference? Is there any difference? So, the point of this is to say, if you do, see, I don't know. One of these is mosquitoes, and one of these is humans, but your mosquitoes are going to be the same. If you do a mosquito-based of those, it's going to get the same risk prediction as human-based ones. Mosquitoes are much easier than humans. You don't have lots of information. So this is, I think, for me, demonstrating that the mosquito-based approach has limits for wings as well. But yes, anyway, so I'll leave from that. Just want to acknowledge that. So my current position is funded from grant, which is jointly funded by Australian Research Council and Department of Agriculture, Fisheries and Forestry, and the CIs there are head grants from NZ and also Lawton and Robert Clark, who's in the room, and Matthew Palliser and Brendan Barnes, who sorry about that. Brendan Barnes from Department of Agriculture, Fisheries, Forestry. Also, in a lot of this infectious disease work, particularly with applications, work with a whole host of other groups, some who's also provided funding over the years. So there's lots of people involved in this work. I have to take lots of questions.
Do we have any questions then? Great, thank you. I was wondering, given your comment about people just use 25 as their standard full sample size, because that's just what you do.
Yeah.
What defaults did you set in your functions when people are using it, or what are you setting as a default in the big version? And what do you find the documentation to combat that?
Yeah, so I avoided setting the defaults. So from the design side of things, I feel like we start with these cost ratios, right? The cost of the different elements of the survey, and from that it calculates, a recommended or sensible survey design. And so rather than having a default size built in, it's as well. You can set it max. So one thing you can say is that these are optimal. These are statistically optimal size, right? Sometimes they're impractical, right? Sometimes you can't fit that many mosquitoes in a tube. A bigger tubes are more expensive. It's not going to do that, or sometimes the sensitivity of the test. The more mosquitoes you've got in there, you're trying to find one worm across the mosquitoes. The more mosquitoes there, you end up sacrificing the sensitivity of the test nodes. You lose some information there. So they might want to put practical action for the biggest. So in the software, people can set like a maximum size, something to set that to 50 by default. But I kind of construct that's putting it the long way. So they have to do a little bit of work, which is, how much work they have to tell the program how much work is it to collect versus test versus process evidence, to avoid that. Yeah, thanks. Okay.
I guess thanks for a great talk. One of your plots you showed optimal pool size against prevalence or loss of plots against prevalence on the axis. But the prevalence to go to zero, pool size didn't asymptote or anything. It just went to a nice finite value.
When the prevalence
tends to zero, so you go on the
yeah. Let me show. Actually, I'm glad you asked this. This is not relevant, but that's a great question. So it does just go to zero. I mean, it's not defined really, but
Well, one of your earlier talk you had one where you had prevalence on the x-axis and it didn't tend to zero, it tended to somewhere for the optimal pool size, not the
optimal pool size. Okay, there's another case. Optimal pool size. Oh, it's the
much better. Yeah, that. So that seems like quite a nice value somewhere around that, because in reality, low prevalence is the difficult problem. Like if your prevalence is 10%, you're going to get quite good estimates and you're going to easily detect it. So it seems to make sense to design on very low prevalence.
Yeah, okay. No, I've got a few questions here. So the optimal pool size, it does add some code, unfortunately, as well. So what I've done here, I've just stopped calculating prevalence of that is too small. Because it just got another infinity. Or in practice, it gets you a maximum pool size. The software will let you calculate. But So yes, it does go up infinity. Yes, I mean on the design effect, what I thought you were talking about is design effect. The design effect doesn't always just sit over one. So one of the funky things, this is almost unique to perfect sensitivity and specificity test, which again, it's not true, they're not better than those two. Yeah, so if the test is not perfectly sensitive, you get roughly what you'd expect from individual testing as well, if the sensitivity is 90%, you miss 10% of the infected people. And so, you lose a little bit of information. So the sensitivity is important, obviously, but from a statistical point of view, it's fairly straightforward. You just have to up your sample size. Specificity gets very it's strange though, so it's sort of counterintuitive results. Essentially, the specificity right there, you're getting false positives. When the prevalence is low, specificity is a killer. Imperfect specificity is a killer because prevalence is 1%, specificity is 99%, which sounds great. But that means half of your positives are true positives and half of them false positives. Right? So it's a real killer to try and estimate prevalence. You've got a nice noise as signal. But with pooling, right? Assuming that the full test doesn't sacrifice much specificity or any specificity, assuming that the full test is just as specific as the individual test. And if we're talking about the later tests, it should be about these things. The genetic signals are usually quite unique, or they're unique by design, right? And so it's other kind of structural sort of things which cause false positives. Anyway, but if we assume that the group test is just as specific as the individual test, you actually gain information by pooling. You don't lose it, you gain information. So this is that slide that I passed over very quickly before. So here's this panel here is the same as the other one. So it's specificity, perfect specificity. And our reference here is individual testing, S equals 1. And the higher the prevalence, the larger the design effect. In other words, the more information you lose. But as soon as you have specificity even slightly less than slightly imperfect specificity, the lower prevalence, because at lower prevalence, so many of the positives will be false positives for individual testing. By group testing, you're reducing the number of total tests you do, so you're reducing the number of false positives, but you're not really reducing the number of true positives, because there's still only so many positives in the population, right? And so your signal to noise ratio get better, and so all of a sudden you can estimate things better if you're doing group testing than if you're doing group testing. And so as a target, the design effect actually goes to 1 less. So for zero prevalence, it's you get 50 times this information from one. No, you get 50 times as much information per unit in a pool size of 50. No, so one pool of 50 is 2,500 times as much information as one individually tested unit. So yeah, so there's a lot more information. So because you've got rid of all these false positives, if that makes sense. So that's another example where group testing might be very beneficial, assuming that you have some characteristics of the test, that the group test is just that specific. Now, I'm not sure how it will affect the boost flow, but that really I think it's cool. So that was a tangent on half a question. You almost asked, and I started answering, we finished asking it. So was there more question?
Yeah, more or less. Yeah, I asked about sensitivity and specificity. How it's affected when you put it all together with this material, your part of your answer there was the genetic thing that it doesn't matter how many you put together, there's going to be the same level of specificity. I know. Try something. But if it's not genetic testing, you know, I've dealt with data where once it gets below a certain dilution level, you fail to detect.
That's right, and the sensitivity is really crushed.
Yeah, and so little testing can have an advantage, but you stay above that level.
That's right, yeah. And I mean, and some of these, I mean, I haven't done teaching this as practical given over for instance, for genetic testing, what they're really having is the number of different, sometimes you have to cycle the machine, the number of times your machine copies the genetic information in your test before the thing that you're interested in is above a certain threshold. So it's kind of all, everything's on one scale, it's all about exponential growth on one scale. And they choose a cutoff based on some gold standard things that we're pretty sure should be negative and things that we're pretty sure should be positive. You do some cutoff accordingly. But even there, there's some fuzziness there. And so if you are changing the cutoff to increase your sensitivity, you might start actually losing specificity. So anyway, so yeah, I think it's hard. And so this is to me, it's feels more like it's a lab question, and unfortunately, people haven't done enough of the sensitivity specificity testing for some of these things. So it's actually slightly unclear what the profile is. We know it works well up to a certain point, but we don't know. Yeah, we don't know how to push it.
Another question in the chat.
Oh. Maybe it's in there.
Well, it's coming too much. Okay, is it the first question?
Yep. Yeah, that's interesting discussion, Dan. Thanks. So yeah, that's one for this question. Yeah, so if just to check that I was correctly, so they talk about there's a trade-off. Obviously, if you're doing pool testing, it takes longer it takes longer to figure out who's positive if you're doing, especially quite complicated pool testing scenarios. So that's not something I have looked at once. So it is so for instance, people have worked more, especially in this sort of Dorfman sort of testing and retesting space. Chris Builder, who's I'm forgetting which university is in the United States, but if I can send him victim shit, has done quite a bit of work on sort of optimizing these design where you do retest individuals or retest the hierarchical structures to do that. And so one of the things that they sometimes optimize for instead of it's I think related to what you're asking, instead of optimizing the number of tests per se, it's the time to the average number of steps per person, things like that. So they're trying to minimize the complexity. But I think perhaps what you're saying is slightly different because if each test is done sequentially, it takes 100 minutes to do 100 tests and only 10 minutes to do 10 tests. If you can minimize the number of tests, perhaps you can still even reduce the amount of time. So perhaps not quite answer your question, but yeah, I think that's a cool suggestion.
We have another one from you.
I mean, any variants of the Dorfman procedure that involve more complex designs, and particularly think of the common genres, like here, or are there any kinds of that would help with pool testing being the number one? Yeah, that's interesting. I mean, yes, there are complicated ones. Again, I'm mostly focused on the case where there isn't retesting. So I can't tell this off my head, I suppose, what there is in terms of common control designs. But certainly, there are different flavors for the array testing, whether you do test the whole lot at once first, or whether you retest all the individual intersections between the rows and columns, or just some of them. So there are some variations there. But yeah, there's lots of variations here. So I think it's sometimes the limit is not what is theoretically optimal you could be used to reduce the number of tests, but what your lab person is willing to put up with in terms of the procedure to go through these things. And so of the ones I'm familiar with, array testing is the most complex that people are willing to do. But there might be, very elegant ones. I mean, for instance, one which kind of helps sometimes. So if you've got, let's say you're just doing regular Northern procedure, you've got a pool of something 100. If the test is positive, you go to the individuals and you start testing them one by one until all the negatives they're clear. You keep on testing until you hit another positive. It's like, great, this could be our one and only positive, right? So that all the rest of them, do we really need to test them? They could be positive still. It could be that there's more than one positive and this is just the first of them, or they could be all negative. So what you do then is you pull everything that comes after that, pull those, and then iterate until you have finally decided for every individual whether or not they're positive or negative. That has some gains over Dorfman, but they're fairly modest. Like the biggest gains are individual versus Dorfman procedure. You massively cut the number of tests, like 80% or whatever. These other ones, they do have gains, but they're sort of increasingly marginal. So, but sometimes that matters if you're trying to screen every child born in a country or a whole host of rare diseases or something like that. So, yeah.
Any other questions here or online? Okay, then thank you again, Marcus McLeod, for being here. And any of us who walk towards the city centre of Canberra for the dinner Quinta. Yeah, so thanks everyone again. Thanks everyone online.
For those who came in a bit later, the attendance books down there, would you please do so? Those meetings for dinner, in time, which is